Hello and welcome everyone to this Epigenetics podcast. In this podcast we're going to talk about DNA packaging, and chromatin architecture. Before we get started, I want to take a step back and think about what we've talked about up to this point in class. Remember, we talked about inheritance. We talked about those basic rules with Mendel. We talked about the extensions to his rules. We talked about the history of inheritance, how we used to think about it, and the way we now are viewing it, and how epigenetics is changing some of the ways we view it. So in that context, we talked a little bit about meiosis. We also spent a decent amount of time talking about transcription, and how with transcription we talked about how we would take this inheritance material, the DNA, and make RNA. And we talked about different kinds of RNA. We spent most of our time talking about messenger RNA, but we also mentioned many other kinds of RNA molecules. And then we talked about epigenetics. Not a lot yet, but enough. We talked about the importance of it, how the rules of epigenetics are changing the way we view inheritance and transcription, and how it's redefining a lot of what we think about genetics. So if you think about it, what we've really done up to this point is we've taken an overview. If this is our cell, and this is our nucleus here, we've looked at the DNA in here. We've thought about how it's inherited. We talked about how these genes here can be transcribed. And when we've been talking about epigenetics up to this point, and just really a kind of a global overview, not the nitty-gritty that we need to get into, but we talked about how epigenetics is making us question, so not really a question mark, but epigenetics is making us really rethink about how these genes are turned on, if they're turned on, if they're turned off, if the process of turning them on or off is inherited, meaning if you turn this gene on or off, is that ability passed on to the next generation of cells or the next generation of the organism. So the big picture is really cool, but how is it all happening? What are these rules of epigenetics? How does this phenomenon of epigenetics help establish what we might call the epigenome? So to really understand it, what we have to do is we've got to dive in here, and we need to start looking at these very specific interactions here. The molecular machinery involved in turning these genes on or off, in packaging the chromosomes, in determining how it's going to be inherited. That just doesn't happen. We want to know the proteins involved that make that happen. And that's what we're going to spend the next several sessions talking about. The first thing we want to talk about is think about how this DNA here is packaged. And the way I drew it here, it looks like it's really easy to get that piece of DNA into this nucleus. But I want to remind you of a few things. We've talked about this before, but in every one of these cells, there's 2 meters of DNA. There's actually more than 2 meters if you count the mitochondrial DNA. But in the nucleus itself, there's 2 meters of DNA in every cell. If you think of the whole organism, like a human for instance, that equates to about 150 trillion meters of DNA, which, if my calculations are right, is about 46 billion miles of DNA in our bodies. In my body, there's about 46 billion miles of DNA. That's insane, I think. That's enough DNA to get all the way out to Pluto and back again. Pluto, I think, how far is that? That's just a few billion miles away. So we could definitely do that. That's how much DNA we have. That's how much DNA we need to get packaged, and we need to figure out how to package it into the cell in a usable way. Another way to look at this is that this nucleus here, this nucleus is about 2 to 10 microns in diameter. The smallest, I'll write that down here, smallest chromosome, human chromosome, is 14,000 times the length of the nucleus. And that's just one chromosome. We got to get them all in there. So let's start trying to understand how that's going to happen. Let's begin this discussion by talking about chromatin. And I'll just draw it here, at least at first, as a DNA molecule. So the first component of chromatin is DNA. Chromatin also contains proteins all around it. Proteins are associated with the DNA for many different reasons. for replication of the DNA, for transcription of the DNA into RNA, for packaging the DNA, which we're going to talk about it quite a bit, and for maintaining the structure. So we have proteins all over this. I'm just going to draw them like this for now, but as you'll see, it's much more organized than that. And I think it would be good to also mention RNA. RNA is a very important part of the chromatin. It sometimes exists just because it's attached to the DNA during transcription, but some RNA molecules play a very critical role in regulating transcription. We have long RNAs, like during X chromosome inactivation. So they play a very important role too. And for now, I'm just going to draw them like this, just to remind us that we have RNA on the chromatin that are very important in maintaining its structure. We're going to spend most of today talking about the DNA and the protein associated with the chromatin. So how is this DNA packaged? Well, at the very preliminary level, it's the double helix, which is just stabilized because of the hydrogen bonds between it. It's not very well packaged at all, and not as usable as we would need it to be. At the next level, we have the nucleosome. And I'm going to put a star here because we're going to spend a lot of time talking about the nucleosome. Mainly because we understand quite a bit about the nucleosome. We understand how it forms for the most part. We understand how it's regulated. We're still trying to understand some of the important dynamics that it has in exactly how it forms. But we do understand it quite well. And we can think of this nucleosome, just for now, we'll get more complex later, is taking this double helix that you see here and wrapping it around these proteins called histones. And because of the way it looks like under an EM, they kind of look like beads on a string, something like that. This nucleosome then is packaged up further into what we call the 30 nanometer form. And though we're beginning to understand the 30 nanometer form a little bit, what we don't really understand is exactly how it forms and the proteins involved. But we have viewed it microscopically. We just don't understand molecularly exactly how it forms. But in essence, what happens here is these nucleosomes begin to package up onto each other. And we do know some of the proteins involved, and we might mention a couple of them, but they can help stabilize this 30 nanometer form. This then gets packaged up even further into a structure we're going to call the 300 nanometer form. We will begin to see this more associated with chromosomes, and we really understand very little about what's happening here. So I'm going to draw it kind of like this at first. And what we think is happening here is that within each of these smaller regions is we have a compressed form, a condensed form of this 30 nanometer form. It then condenses more to about the 700 nanometer form, and it's just a further condensation of this 300 nanometer form. And then ultimately, what we'll see here at some point is the mitotic chromosome. Anytime you've looked at a karyotype and you see all those chromosomes on them, that's what we see here, the fully condensed form of the packaged DNA. For this podcast, and probably the next one, it'll probably take us 2 to get through it, is we're going to focus on these 2 forms here, the nucleosome and the 30 nanometer form, and mainly the nucleosome. So let's dive in here and begin to appreciate how these proteins here are binding to the DNA, or rather the DNA is binding to the proteins, I might. So let's talk about this nucleosome. We'll talk a lot about the nucleosome. It will soon become one of your dear friends. I'm going to first draw the nucleosome as best as I can here. It contains 8 proteins. It contains 2 proteins that we're going to call H3. And there's one behind this one that I'm just not drawing for simplicity. There's another protein called H4. And there's another H4 behind this. There's 2 of each of these. And then there's another histone that we're going to call H2B. And there's another H2B right behind it. And lastly, I'm going to show you here H2A. And not surprisingly, there's another one right behind here. So it exists as the nucleosome contains these 8 proteins, these 8 histone proteins, so we call it an octamer. And the DNA will come along here and it'll wrap around this octamer. I'm going to draw a neighboring one here, and it will wrap around this one. And if I didn't show it precisely, the amount of DNA that is wrapping around each of these is very specific. It's not as random as I may have drawn it here due to my limited drawing skills. I'm going to talk about the DNA a little bit here, well actually quite a bit, but I'm going to just open it up with talking about the DNA that's associated with the nucleosome. And to be clear, when I say nucleosome, this is what I'm talking about. The piece of DNA that's being held by the proteins. The nucleosome is the octamer plus 147 base pairs of DNA. Now this linker DNA, the DNA in between these two nucleosomes, can be up to 80 base pairs. It's usually not quite that much, but it can be. Another term I'm going to bring up here is this distance that we refer to from the center of one nucleosome to the center of the other nucleosome. And we call that the nucleosome repeat length. And the cool kids call it NRL. Now there's one more protein I would be remiss if I did not introduce you to. It's a very interesting one. It's also a histone protein. It doesn't share the same level of homology with these other ones. And in fact, its importance has often been disputed. And its existence in some eukaryotes has been disputed. And it's called H1. And its job is to link these two neighboring nucleosomes. We think it stabilizes their interactions and plays a role in further packaging of the chromatin beyond the level of the nucleosome as we move into the 30 nanometer form. We do believe it to be important because, and we'll talk more about why we think it's important later on. Now I want to spend a little bit more time talking about these histone proteins. When we compare histone proteins, so let's just write H2A, H2B, H3, and H4. We'll leave H1 out of this. Structurally, they're similar, both within a species and between species. However, by sequence of the genes that code these proteins and the protein sequence, there's large differences, suggesting that the histone proteins might be able to bind DNA in general, but not not sequence specific. That is, maybe the histone proteins don't recognize a very specific sequence. Rather, maybe they just recognize DNA and they're able to bind to DNA. One thing that came out of the X-ray crystallography studies is this motif that is shared by all of the histone within a species and between species, called a histone fold. This histone fold is very important in dimerization. Remember, there are two H2As, two H2Bs, two H3s, two H4s. The ability to form those homodimers of each of those seems to be dependent upon these histone folds. Though I'm never going to make you draw this on a quiz or a test, I think it's interesting just to look at this structure. Essentially, it forms this really long helix, like so, and then it loops around and forms this other helix, something like that. It folds there, so it's a helix fold. And then over here, there's another fold with another helix. So there are three alpha helices, one long central one with these two on either end that kind of form this bend or a fold, which is how it got its name. Okay, one more thing I want to mention about these histone folds, which is really amazing, is how well they are conserved. We see these in archaea and throughout eukaryotes. And to remind you, we look at a tree here. We'll have our bacteria right here. We'll have our archaea here. And we'll have our eucs here. There's been hundreds of million years between the eucs and the archaea. Yet, this histone fold motif has been well conserved throughout that time with very few changes. Now, archaea don't have nucleosomes in the same way that eukaryotes do, but they do have histone-like proteins that likely function in the formation, in interacting with DNA in some packaging mechanism. That is one of the many similarities that archaea have with eukaryotes. Okay, the last thing I want to mention about these histone folds is that in addition to help forming dimers, they also play a role in binding DNA. And I'm going to erase what we have here so I can talk about that in more detail next. Okay, I'm going to draw the nucleosome again here with DNA wrapping around it. And I mentioned that these histone folds are important in forming the dimers with their same counterparts, which we can't see because they're behind here. But I just said that they play an important role in binding the DNA. And though I'm not sure if it's apparent from this drawing, but you could look in your book and you'd see it represented a lot better, is that the DNA doesn't go in between through the core of the histone proteins. Rather, it wraps around it. So histone folds, not going to really be able to draw it, but I'm going to indicate it. They're shown on the, and I'm going to just draw it like so, HF, HF. They're on the outer side of the histone protein. They're not tucked up in the middle of the protein, so they contain some amino acid residues that position them on the outside of the protein. On the outside of the protein, through crystallography studies, we know that these histone folds interact directly with the DNA. Now, if you go way back in your genetics class, you may remember these terms called major grooves and minor grooves. And at the time, if you were like me when I was in genetics, I didn't think much of them. I just thought of them as components of the DNA structure, and they just formed because that's the way the helix formed, and they were just stuck that way. But it turns out they're incredibly important in positioning certain proteins. To help illustrate what these major and minor grooves look like, I want to show you this picture because it will be nearly impossible to draw. Over here we see the double helix, and you can see as it wraps around here we form this much more narrower groove between the phosphate backbones, and then here we have a much larger groove between this phosphate background and that phosphate background, and they alternate minor, major, minor, major. And I won't ask you, I promise I won't ask you the lengths of these, the dimensions, how many angstroms they are. That isn't important. But what is important is that you know that these two grooves exist, the larger one, the major groove, and then the smaller one, the minor groove. And you can imagine now, because of these sizes and because of the fixed sizes, that proteins can bind specifically based on the sequences, because here's our base sequences here, but also proteins can bind in here because they can physically wedge in between either the major or the minor grooves. Many DNA binding proteins have specific binding spots either in major or minor grooves. Okay, I'm going to move this picture over here just a little bit because I want to draw some things here about this histone fold. So the DNA that wraps around these histone proteins and comes in contact with these histone folds. There are 14 minor grooves in this region, just like these that you see here. Of those 14, 12 of them come in contact with this histone fold. Let me move these up here, minor groove, and I'm not going to write down the major groove because the histones don't bind to the major grooves. So 12 of the 14 minor grooves bind to the histone fold, which I'm just going to abbreviate as HF. In addition to the DNA binding right into these minor grooves, we know there are other contributions that can't be ignored in maintaining the stability of the structure. So other contributions between histone and DNA are important in maintaining stability. To talk about these other contributions, I want to point out that the binding of DNA to histone proteins, so as the DNA wraps around here and binding to these histones, this binding is not equal at all locations. Meaning there are parts of this interaction between DNA and histone that bind tighter and other parts that bind with less strength. Probably the most important interaction that leads to the strength of this binding are electrostatic forces, or let me say bonds, like ionic bonds. More specifically, there are positive residues, positive amino acids on these histone folds that play a role in these ionic bonds with the DNA. So positive amino acids on the histone fold. So here are lots of abbreviations so I can fit this in here. Positive amino acids on the histone folds bind through these electrostatic interactions. So they bind with DNA. And remember, DNA is largely negatively charged because of the phosphate backbone. So I'm going to write up here positive amino acids. And these would be like lysines and arginines bind with the phosphates of DNA at very specific regions. And it's not just one lysine or arginine. Another important thing to point out is that there are negative amino acids on the opposite side. Meaning these negative amino acids, like glutamic acid and aspartic acid, are not near the phosphates. So they don't interfere with this interaction. I want to circle this right here, because this turns out to be incredibly important when we move on to the next chapter and beyond, when we talk about regulating gene expression by removing these. And so keep that in mind. So keep that somewhere fresh in your brain, because we're going to keep coming back to that. Another interesting observation is that since this association between these histone amino acids and the DNA molecule occur on the DNA phosphate backbone and not on the nucleotide sequence, this suggests that this association is not sequence specific. Since it's with the phosphate backbone, one might assume these histones could form nucleosomes on any stretch of DNA in a sequence independent manner. Another point I want to make is that bending of this DNA is very distorted. We might even say it's energetically costly. It takes a lot of energy to distort the DNA in this manner and to maintain it that way, which suggests that these interactions that hold this all together, that these interactions are very strong to maintain this quite distorted state of the DNA. Okay, I'm out of room, so I need to erase some of this, but I'm still want to talk more about how these histones are binding. As I said previously, this binding of the histones to the phosphate backbone leads us to think this is probably not a sequence-specific interaction. Though there are some who say that, and there's some evidence to support this, that this interaction with the DNA phosphate backbone provides kind of a general binding, meaning that this interaction is really important to get the histones here and get them in the proper position, but perhaps there is a sequence specificity that sort of fine-tunes the positioning of the histones. What supports this is the fact that when they do X-ray crystallography studies and they examine the structure here, there are several hydrogen bonds that could form between histones and DNA bases, where the histones could form these hydrogen bonds with some of the hydrogens that are found on the bases of the DNA sequence. So at this point, this is more speculative, but there is some growing evidence to support that. Now I want to talk about how this actually forms. So in thinking about how they might form these nucleosomes, the histones in the DNA, early on scientists took DNA plus histones and mixed them in a test tube, and they got nucleosomes. This suggested that this was a spontaneous event. But was it? What they discovered was if they added to this mix here oocyte cytoplasm, and they took it from xenopus, frogs, and they added that cytoplasm to this reaction, what happened was this occurred at a much faster rate. This went on to suggest then that they could form spontaneously outside of the cell, but more than likely within the cell there were factors found in the cytoplasm that would speed up this reaction. And this led to the discovery of many histone chaperone proteins. If you remember from your cell biology class, chaperones help fold proteins to their three-dimensional shape. So these histone chaperones play a role in not only folding these proteins, but helping them form these histone-histone interactions. there's evidence to show that these histones come onto the DNA in a stepwise manner, and that the chaperone, these histone chaperone proteins, has specificity to only a subgroup of histones. So they don't just bind any histone, they have to bind specific histones. So at present, we suspect these histone chaperones are very important in setting up this nucleosome structure. The mechanism that it accomplishes this feat is currently unknown. Okay, that ends what I'm going to talk about for this first podcast. In this podcast, we talked about the nucleosome structure. We talked about the histone proteins, how the histone proteins interacted with the DNA to form the nucleosome. We talked about very specific features of the histone proteins that allowed for this interaction. In the next podcast, what I want to talk about is how these histones, rather these nucleosomes, now help form the next level of chromatin structure, the 30 nanometer form.